Good morning, everyone, and welcome to today's presentation. I'm Adil Singh, and I hold a PhD in microbiology from IIMT University, Meerut. Today, we're going to explore the fascinating emergence of microbiology as a distinct scientific discipline. Over the course of this presentation, we'll look at how microbiology developed, key milestones, and its impact on science and society. I'm excited to take you through this journey, so let's get started. This slide doesn't contain enough information. That's why we're moving to the next slide. Acharya Sushruta was an ancient Indian physician and surgeon whose contributions have had a lasting impact on medicine. He lived around 600 BCE, during the Vedic period in India, a time when many foundational aspects of Indian culture and knowledge were developing. He is widely regarded as the father of Indian surgery, a title that speaks to his pioneering work in the field. His most famous work, the Sushruta Samhita, is one of the most important surviving ancient treatises on medicine and surgery. This extensive text is a massive compendium that covers all aspects of general medicine, but what really sets it apart are the detailed descriptions of surgical procedures. It's incredible to think that such a comprehensive guide existed thousands of years ago. Sushruta was a true pioneer, especially in the area of surgical techniques. He is credited with developing early forms of rhinoplasty, which is nose surgery, as well as cataract surgery and even cesarean sections. These innovations highlight his advanced understanding of anatomy and his inventive approach to treating patients. His work didn't just influence India, it had a global impact. His surgical techniques and medical knowledge were adopted by physicians in the Middle East and Europe, helping to shape the development of modern surgery. Today, his work continues to be studied by medical students around the world, underscoring the timeless relevance of his contributions. In essence, Acharya Sushruta's legacy is a testament to the rich history of Indian medicine and its role in advancing human health and surgical science. Let's take a closer look at some of Sushruta's incredible contributions to medicine and surgery. He described more than 180 surgical procedures, which is astonishing, and over 100 surgical instruments, showing his deep understanding of the tools and techniques needed for successful operations. Interestingly, he was among the first surgeons to use anesthesia and antiseptics, which significantly improved patient safety and comfort. He also developed innovative techniques for procedures like rhinoplasty, which is nasal reconstruction, cataract surgery for vision problems, and cesarean sections for childbirth. His work wasn't just about performing surgeries. He also wrote extensively on treating wounds, fractures, and other injuries, laying a foundation for trauma care. Beyond surgery, Sushruta made important contributions to public health. He emphasized sanitation and hygiene, recognizing their vital role in preventing disease and promoting overall health. All of these achievements highlight how Sushruta's work laid the groundwork for many modern medical practices, connecting surgical innovation with public health efforts. The golden age of microbiology is a fascinating period in history, generally considered to span from the mid-1800s to the early 1900s, roughly between 1857 and 1914. During this time, we saw a flurry of groundbreaking discoveries that fundamentally changed our understanding of microorganisms. These advances helped us understand their role in causing diseases, food spoilage, and other crucial biological processes. Now, let's talk about some of the key figures from this era. First, there's Louis Pasteur, a French chemist and microbiologist. He's best known for his work on germ theory, which proved that microorganisms are the cause of many diseases. Pasteur also developed pasteurization, a process to kill bacteria in food and beverages, and he was instrumental in creating vaccines for diseases like rabies and anthrax. Then there's Robert Koch, a German physician and microbiologist. He made significant contributions by identifying the specific causes of diseases such as anthrax, tuberculosis, and cholera. Importantly, Koch developed a set of criteria, what we now call Koch postulates, that help scientists establish a direct link between a microorganism and a disease. Together, these scientists helped establish microbiology as a rigorous scientific discipline. Their work laid the foundation for modern medicine and public health, connecting directly to our overall theme of understanding and controlling infectious diseases. Let's take a closer look at some key figures in the history of microbiology. First, there's Ferdinand Kohn, a German botanist and microbiologist. He's well known for his work on classifying bacteria and algae, which helped lay the groundwork for understanding microbial diversity. Kohn also developed important techniques for cultivating and studying microorganisms, making it easier for scientists to observe and analyze them in the lab. Next, we have Antony van Leeuwenhoek, a Dutch tradesman and microscopist. He's often called the father of microbiology because he was the first person to observe and describe microorganisms with a microscope. His pioneering work opened up a whole new world of tiny life forms that we couldn't see before, fundamentally changing our understanding of biology. And then there's Joseph Lister, a British surgeon. He's famous for his work on antiseptic surgery. Lister was the first to use carbolic acid to sterilize surgical instruments and dressings. This innovation significantly reduced the risk of infections during surgery, greatly improving patient outcomes and establishing the importance of sterilization in medicine. All of these scientists played crucial roles in advancing microbiology, each contributing vital methods and discoveries that connect to the broader theme of how understanding microorganisms has transformed medicine and science. Let's take a closer look at some of the most important discoveries during the golden age of microbiology. Starting in 1857, Louis Pasteur made significant strides with his work on fermentation, which laid the groundwork for understanding microbial activity in food and beverages. Just a few years later, in 1861, he challenged the idea of spontaneous generation, demonstrating that microbes come from other microbes, not from non-living matter. In 1864, Pasteur developed pasteurization, a method to kill harmful bacteria in liquids like milk and wine, which has had a huge impact on public health. Moving forward to 1867, Joseph Lister introduced antiseptic techniques in surgery, drastically reducing infections and mortality rates. Then, in 1870, Ernst Karl Abbe improved microscopy with his condenser and oil immersion objectives, allowing scientists to see microbes more clearly. In 1876, Robert Koch proposed the germ theory of disease, establishing that specific microbes cause specific diseases, which was revolutionary for medicine. That same year, Neisser identified Neisseria gonorrhoeae, the bacteria responsible for gonorrhea, adding to our understanding of infectious agents. In 1880, Pasteur developed immunization techniques, paving the way for vaccines. The following year, Robert Koch achieved the first pure culture of bacteria, enabling precise study of pathogens. Also in 1882, Koch identified Microbacterium tuberculi, the bacteria responsible for tuberculosis, and Walter Hess introduced agar as a solid medium for growing bacteria, which became a standard in microbiology labs. Finally, in 1883, Koch identified Vibrio cholerae as the causative agent of cholera. These discoveries collectively marked a golden era in microbiology, transforming our understanding of microbes and disease and setting the stage for modern microbiological science. In 1884, we see some remarkable discoveries that really laid the groundwork for modern microbiology. Eli Machnikov identified phagocytosis, the process by which cells ingest harmful particles, which is fundamental to our immune defense. At the same time, H. Christian Graham developed the Graham staining technique, a crucial method for classifying bacteria and guiding treatment decisions. Also in 1884, Escherich contributed to identifying Escherichia coli, a bacterium now central to both microbiology and medicine. Moving forward to 1887, Petri invented the Petri dish, a simple yet revolutionary tool that allowed scientists to culture bacteria in a controlled environment. Then, in 1890, von Behring made a breakthrough with the diphtheria antitoxin, which was among the first effective treatments for bacterial diseases. That same year, Paul Ehrlich proposed the theory of immunity, suggesting that our bodies could produce specific defenses against pathogens. By 1892, Winogradsky was exploring the sulfur cycle, highlighting the importance of microbes in biogeochemical processes. In 1898, Shiga discovered Shigella dysenteriae, the bacteria responsible for dysentery, further advancing our understanding of infectious disease agents. In 1910, Paul Ehrlich, often called the father of chemotherapy, made significant strides in developing treatments for diseases like syphilis. These discoveries, spanning from cellular processes to disease treatment, collectively underscore how much microbiology evolved during this period, building the foundation for the advanced immunology and microbiology we study today. Joseph Lister, who lived from 1827 to 1912, was a prominent figure in the history of surgery. He served as a professor of surgery at Glasgow Royal Infirmary, where he made groundbreaking contributions. Lister applied Louis Pasteur's work on microorganisms and fermentation to the field of surgery. In 1867, he introduced antiseptic techniques that dramatically reduced infections in surgical wounds. This was a major milestone because it marked the shift from the old belief in laudable pus as part of healing to modern aseptic practices focused on preventing infection altogether. Because of his pioneering work, he's often called the father of modern surgery and antiseptic surgery. His key innovation involved using carbolic acid, or phenol, to sterilize surgical tools and clean wounds. This approach significantly lowered the rates of wound sepsis and gangrene, which in turn reduced the need for amputations. Lister's contributions truly transformed surgical practice and saved countless lives. His work laid the foundation for safer, cleaner surgeries that continue to save lives today. So, what did Lister want to achieve? His goal was to decrease the rates of wound sepsis that frequently occurred in hospital settings. He was particularly intrigued by the work of Louis Pasteur, which he learned about through Thomas Anderson, a chemistry professor in Glasgow. Pasteur had shown that liquids like milk and juice had much lower rates of fermentation and putrefaction when protected from the air. From this, he concluded that airborne microbes were responsible for these processes. This was a groundbreaking discovery because it disproved the popular theory at the time that harmful microorganisms could generate spontaneously in these liquids. Lister took this idea and hypothesized that microorganisms in the air were also causing wound infections and suppuration. At that time, there was no clear way to differentiate between harmful and harmless microorganisms, but Lister believed strongly that the air in medical environments needed to be disinfected to prevent infection. He argued that in the post-surgical setting, infections could be avoided if we placed an antiseptic barrier between the wound and the surrounding air. This idea was quite revolutionary. It was even thought that many diseases might be prevented entirely by removing or killing airborne germs, whether through filtration, heat, or chemicals. And that's how Pasteur's work directly influenced Lister's approach, highlighting the importance of controlling airborne microbes to improve surgical outcomes and reduce infections. Lister's most important contribution to medicine was his development of antiseptic surgery. He was the first surgeon to systematically apply the principles of germ theory to the practice of surgery. He recognized that germs were the cause of wound infections, and that these infections could be prevented by killing the germs before they entered the wound. Lister experimented with a variety of antiseptics, but he eventually settled on carbolic acid, also known as phenol. He developed a series of procedures for using this chemical to sterilize surgical instruments, dressings, and even the hands of the surgeon. Additionally, he used carbolic acid to spray the air around the patient during surgery. This antiseptic system proved to be highly effective in reducing wound infections. In fact, in one study, Lister found that the mortality rate for patients with compound fractures dropped from 45% to just 15% after he started applying antiseptic techniques. Overall, Lister's work was a groundbreaking step forward in surgery and infection control, and it really laid the foundation for modern aseptic techniques that we still rely on today. Lister's work truly revolutionized the field of surgery. Thanks to his innovations, procedures that were once too risky or impossible became feasible and much safer. Because of this, he's considered one of the most important figures in the history of medicine. But his contributions didn't stop there. Alongside his work on antiseptic techniques, Lister also made significant advances in physiology and ophthalmology. He conducted pioneering research on the blood vessels of the eye and the nervous system, which helped deepen our understanding of these vital systems. Additionally, Lister invented several surgical instruments, including the famous Lister's scissors and the Lister's bandage, which improved surgical practices and patient outcomes. His impact on medicine has been profound, saving countless lives and making surgery safer and more effective for millions worldwide. His legacy really underscores how innovation and dedication can transform healthcare and improve human well-being. Louis Pasteur was a French chemist and microbiologist who is widely regarded as one of the most important founders of microbiology. His work essentially helped shape the scientific discipline of microbiology into what it is today. Interestingly, he was the first to coin the term microbiology, specifically for the study of tiny organisms that are invisible to the naked eye. Because of his numerous groundbreaking contributions, he's often called the father of modern microbiology. Pasteur is renowned for discovering key principles that still influence medicine and science, such as vaccination, microbial fermentation, and pasteurization. These discoveries not only advanced our understanding of microorganisms, but also led to practical methods for preventing and controlling diseases. He is particularly remembered for his remarkable breakthroughs in understanding the causes of diseases and how to prevent them, which has saved countless lives. Finally, it's important to note that he is regarded as one of the three main founders of bacteriology, alongside Ferdinand Cohn and Robert Koch. Together, these scientists laid the foundation for modern microbiology and infectious disease research. So, as we see here, the studies on fermentation sparked Louis Pasteur's interest in microbiology. This led to some groundbreaking contributions that truly changed the field. For instance, Pasteur disproved the long-held theory of spontaneous generation of disease. He showed that microorganisms are not generated from nothing, but actually come from existing microbes. He then postulated the germ theory of disease, which states that microorganisms are the cause of many illnesses, instead of bad air or vapors as previously believed. His experiments were key. They demonstrated that without contamination, microorganisms simply wouldn't develop. This was a major breakthrough in understanding how infections spread. Moving on, Pasteur also proposed principles for food preservation through fermentation, applying microbiology to practical uses. He introduced sterilization techniques, developing devices like the steam sterilizer, hot air oven, and autoclave, which are still vital today in medical and food industries. He also described the process of pasteurization, particularly for milk and wine, which greatly improved safety and shelf life. Additionally, Pasteur's work significantly reduced maternal mortality from puerperal fever, a bacterial infection following childbirth. Beyond these, he contributed to vaccine development for diseases such as anthrax, chicken cholera, and rabies. All of these achievements highlight how Pasteur's work laid the foundation for modern microbiology and public health, showing us the power of science in improving human lives. Pasteur's development of the vaccine for hydrophobia, or rabies, was truly a milestone in the history of medicine. It marked a significant breakthrough in immunization, helping to protect countless lives from this deadly disease. Moving on, let's talk about his liquid media concept. Pasteur used nutrient broth as a medium to grow microorganisms, which was a revolutionary idea at the time. This approach allowed scientists to cultivate and study microbes more effectively, laying the groundwork for modern microbiology. He was also the founder of the Pasteur Institute in Paris, which remains a leading center for research in infectious diseases and immunology today. But Pasteur's contributions weren't limited to microbiology. In chemistry, he made important discoveries, particularly regarding the molecular basis for the asymmetry of certain crystals and the phenomenon of racemization. Early in his career, he investigated tartaric acid, which led to the first resolution of what we now call optical isomers. This work was crucial because it helped us understand how molecules can have the same chemical composition but differ in their structure and properties. His research paved the way for our current understanding of the fundamental principles in organic chemistry. So, overall, Pasteur's work bridges microbiology and chemistry, illustrating how interconnected scientific disciplines can lead to groundbreaking discoveries. Louis Pasteur's swan neck flask experiment was a series of groundbreaking experiments conducted in the 1860s, aimed at disproving the long-held theory of spontaneous generation. This theory suggested that life could arise spontaneously from non-living matter, which we now know isn't true. In his experiment, Pasteur used a specially designed flask with a long, curved neck that resembled a swan's neck. He filled this flask with nutrient-rich broth and then boiled it to kill any microorganisms that might already be present. The boiling sterilized the broth, ensuring that any future growth would come from outside sources. After boiling, he left the flask open to the air, but the unique curved shape of the neck prevented dust, particles, and microorganisms in the environment from falling into the broth. This design was crucial. Pasteur observed that the broth inside the flask remained clear and free of microorganisms for weeks, despite being exposed to air. This was a key point, because it showed that microorganisms do not spontaneously generate from non-living matter, but rather come from external sources like dust or microorganisms in the air. To test this further, Pasteur broke off the curved neck of one of the flasks. This change allowed dust and other particles from the air to fall directly into the broth. Within just a few days, the broth turned cloudy, and microorganisms began to grow. This demonstrated clearly that the microorganisms came from the environment, not spontaneously from the broth itself. This experiment was pivotal because it provided strong evidence against spontaneous generation. It showed that life comes from existing life, supporting the idea of germ theory and helping us understand the true origins of microorganisms. It was a major step forward in microbiology and scientific thinking overall. Pasteur's experiment was truly groundbreaking because it demonstrated that microorganisms come from other microorganisms, not from non-living matter. This was a huge shift in understanding, as it challenged the idea of spontaneous generation. Instead, he proved that these tiny life forms have a source, another microorganism, that produces them. This insight was crucial in establishing the germ theory of disease, which links specific microorganisms to specific illnesses. Now, the swan neck flask experiment is one of the most famous scientific experiments ever conducted. It's a perfect example of scientific elegance, simple in design, but powerful in its implications. What makes it so impactful is how it visually and practically demonstrated that microbes do not spontaneously appear from nothing, but rather come from other microbes in the environment. Overall, these experiments helped us understand the fundamental nature of microorganisms, paving the way for advances in microbiology and medicine. They underscore the importance of careful scientific methodology and open the door to controlling and preventing diseases by understanding their microbial origins. Conti is a crucial part of our discussion because it really highlights the interconnectedness of the concepts we've been exploring. When we talk about Conti, we're focusing on its role in shaping the overall strategy and how it influences the decision-making process. It's important to understand that Conti isn't just a standalone element. It ties into the bigger picture of how we approach challenges and opportunities. By examining Conti closely, we can identify key patterns and frameworks that help us stay aligned with our goals. This ties back to the overarching theme of integration and strategic thinking throughout this presentation. So, as we move forward, keep in mind how Conti exemplifies these principles in action and why it's so vital to our success. Antony van Leeuwenhoek was a Dutch scientist and tradesman, often regarded as the father of microbiology and the father of microscopy. He was the first scientist to observe microbes, which was a groundbreaking discovery at the time. His pioneering work in microscopy and his contributions to the discovery of microorganisms really laid the foundation for modern microbiology. He was born in Delft, Netherlands, in 1632, and he lived until 1723. Leeuwenhoek is best known for designing and building simple yet incredibly powerful microscopes. These microscopes allowed him to observe a wide variety of microorganisms, bacteria, protozoa, even blood cells, for the first time in history. What's fascinating is that Leeuwenhoek crafted some of the most powerful microscopes of his era, with lenses that could magnify objects 200 to 300 times. This level of magnification was extraordinary at the time and opened up a whole new world of tiny life forms that we now understand to be fundamental to biology and medicine. So, Leeuwenhoek's work not only advanced science, but also demonstrated how innovation and curiosity can lead to profound discoveries that change our understanding of the natural world. A.V. Leeuwenhoek was the first person to observe and document single-celled organisms, which he called animalcules. His detailed observations of bacteria challenged the long-held belief in spontaneous generation, providing strong support for the idea of biogenesis, the concept that living things come from other living things. He didn't stop there. He also examined human cells, including blood cells and sperm cells, which helped us understand human anatomy at a microscopic level. This was a huge step forward in biology and medicine. Leeuwenhoek received support and recognition from important institutions like the Royal Society in London. His letters, where he described his fascinating discoveries, caught the attention of leading scientists of his time. This gave him a platform to share his findings and helped validate his work. Most importantly, Leeuwenhoek's discoveries played a key role in developing the germ theory of disease. This theory established the link between microorganisms and illnesses, revolutionizing medicine and leading to major advancements in public health practices. All of these contributions show how his pioneering work laid the foundation for modern microbiology and transformed our understanding of the microscopic world. His observations of cells and microorganisms provided crucial evidence for the development of the cell theory. This theory is one of the fundamental principles of biology, emphasizing that all living organisms are made up of cells. Essentially, whether you're looking at a tiny microorganism or a complex human body, the building blocks are the same: cells. This discovery helped scientists understand that life's diversity is built upon a common cellular foundation, connecting all living things in a remarkable way. It also marked a significant step forward in our understanding of biology, illustrating how microscopic observations can lead to broad, universal principles. Robert Koch was a prominent German physician and microbiologist who made significant contributions to medical science during the late 19th and early 20th centuries. He was born on December 11, 1843, and passed away on May 27, 1910. He is widely recognized as one of the founders of modern bacteriology, a field that revolutionized our understanding of infectious diseases. Koch identified the specific causative agents of diseases such as tuberculosis, cholera, and anthrax, which was a breakthrough in linking microbes to specific illnesses. He also provided experimental support for the concept of infectious disease by conducting experiments on both humans and animals, which helped solidify the germ theory of disease. Because of his pioneering work, he is regarded as a key figure in public health, influencing legislation and shifting attitudes toward hygiene to prevent the spread of infections. His work on tuberculosis was particularly impactful, earning him the Nobel Prize in Physiology or Medicine in 1905. Overall, Koch's discoveries laid the groundwork for how we diagnose, treat, and prevent infectious diseases today. In 1876, I investigated the disease cycle of anthrax, which helped us understand how this disease spreads and persists. Then, in 1882, I studied the bacteria responsible for tuberculosis, and a year later, in 1883, I focused on cholera. Through this work, I identified important bacteria such as the anthrax bacilli, tubercle bacilli, and cholera bacilli, which are crucial to understanding these diseases. One of the key concepts I observed was the phenomenon of acquired immunity, which explains how exposure to pathogens can lead to protection against future infections. To advance bacterial research, I introduced solid media for cultivating bacteria, developing the pour plate method, and becoming the first to use solid culture media for this purpose. These innovations greatly improved our ability to isolate and study bacteria precisely. I also developed methods for isolating bacteria in pure cultures, which is essential for accurate identification and understanding of pathogens. Additionally, I described the hanging drop method, a technique for testing bacterial motility, helping us understand how bacteria move. Staining techniques using aniline dye were another breakthrough I introduced, allowing clearer visualization of bacteria under the microscope. I also invented the hot air oven and steam sterilizer, which are vital for sterilization processes, and I devised methods to assess the effectiveness of antiseptics, ensuring better infection control. Overall, my contributions laid the foundation for microbiology as a scientific discipline, enabling us to identify, isolate, and combat infectious diseases more effectively. Let's take a closer look at Koch's postulates, which are a fundamental set of rules for linking a specific microorganism to a disease. First, the microorganism must be found in all individuals affected by the disease, but crucially, it shouldn't be present in healthy people. This helps us establish a strong association between the microbe and the illness. Next, we need to be able to isolate that microorganism from a diseased individual and grow it in a laboratory culture. This step is vital because it allows us to study the microbe in detail and confirm it's the same one responsible for the disease. Then, when we introduce this cultured microorganism into a healthy host, it should cause the same disease. This step provides evidence that the microbe is capable of producing the illness under controlled conditions. Finally, after the disease develops in the healthy host, we re-isolate the microorganism and verify that it's identical to the original. This confirms the causative role of that microorganism in the disease process. Overall, Koch's postulates help us establish a clear cause and effect relationship between microbes and diseases, which is essential for understanding and controlling infectious diseases. Conti, as we approach the final stages of our project, it's crucial to focus on the aspects that will ensure a successful completion. This includes reviewing our key deliverables, making sure all deadlines are on track, and confirming that our team is aligned on the next steps. Remember, maintaining clear communication throughout this phase is vital to avoid any misunderstandings or delays. By staying organized and proactive, we can smoothly transition into the closing phase and set the stage for future opportunities. So, let's keep up the momentum and finish strong. Edward Jenner, who lived from 1749 to 1823, was an English physician and scientist. He is widely credited with developing the smallpox vaccine, which was a huge breakthrough in medicine. Because of his pioneering work in vaccination, he is often called the father of immunology. The terms vaccine and vaccination actually come from the Latin word varioli vaccine, which means pustules of the cow. Jenner himself coined this term to describe cowpox. In 1798, he used it in the title of his famous inquiry into the varioli vaccine, which he called the cowpox. In this work, Jenner described how cowpox could protect against smallpox. Now, a key experiment that really demonstrated this was done in 1796. Jenner inoculated an 8-year-old boy named James Phipps with pus from a cowpox lesion. Phipps developed a mild case of cowpox, but he recovered quickly. Then, 2 months later, Jenner inoculated Phipps with smallpox pus. And here's the remarkable part: Phipps did not develop smallpox. This proved that cowpox could be used to vaccinate people against smallpox, which was a deadly disease at the time. So, through Jenner's work, we see the foundation of modern immunology and vaccination. His discovery paved the way for the eventual eradication of smallpox and has saved countless lives. Jenner's smallpox vaccine played a crucial role in the global effort to eradicate smallpox. Mass vaccination campaigns using Jenner's innovative method significantly reduced the incidence of the disease. Over time, these efforts led to the complete eradication of smallpox in 1980, which remains one of the greatest achievements in public health. This milestone serves as a powerful testament to how effective vaccination programs can be when based on pioneering work like Jenner's. In summary, Edward Jenner's development of the smallpox vaccine revolutionized medicine and immunology. It not only led to the eradication of smallpox, but also paved the way for the widespread use of vaccines to prevent many other infectious diseases. Ultimately, his work has saved countless lives around the world, highlighting the profound impact of scientific innovation in improving global health. D. Ivanovsky was a Russian botanist and microbiologist who is credited with the discovery of viruses in 1892. He was studying a disease called tobacco mosaic disease, which caused tobacco leaves to discolor and become mottled. Ivanovsky used a filter to remove bacteria from the sap of infected plants, expecting that this would stop the disease. However, the filtered sap was still able to infect healthy plants, which was quite a surprising finding at the time. This led him to the conclusion that the disease was caused by an infectious agent smaller than bacteria, something that could pass through the filter. Ivanovsky's work was groundbreaking because it introduced the idea that there are infectious agents even smaller than bacteria, which we now know as viruses. His contributions to virology have had a profound impact on our understanding of infectious diseases. Today, we know that viruses are responsible for a wide range of illnesses, from the common cold to more serious diseases like HIV/AIDS. Ivanovsky's discoveries also paved the way for the development of vaccines and other treatments for viral infections. If we look at the structure of the tobacco mosaic virus, it's a tiny hollow rod made up of a single spiraling strand of RNA surrounded by a protein coat. This discovery helped us understand the basic architecture of many viruses. Dmitry Ivanovsky, who lived from 1864 to 1920, truly revolutionized our view of infectious agents, and his work remains a foundational part of virology today. M.W. Beijerinck was a Dutch microbiologist and botanist, and he's widely regarded as one of the founders of virology. He's credited with discovering viruses, which he originally called contagium vivum fluidum, meaning contagious living fluid. In 1898, he conducted filtration experiments that showed tobacco mosaic disease was caused by an infectious agent smaller than bacteria. This was a groundbreaking discovery because it expanded our understanding of infectious agents beyond bacteria. Beijerinck was also a pioneer in environmental microbiology. He invented the enrichment culture method, a technique still used today to isolate and study microbes from the environment. His work helped us understand critical processes like nitrogen fixation and sulfur cycling, which are essential to life on Earth. Overall, Beijerinck's contributions laid the foundation for many areas in microbiology and environmental science, connecting the dots between microscopic agents and global ecological processes. His work truly transformed our understanding of microbes and their role in nature. Now, let's talk about some of Beijerinck's most important discoveries. First, he studied viruses using the tobacco mosaic virus, or TMV. This was a groundbreaking moment because he was the first to realize that viruses are actually reproducing entities, quite different from other organisms like bacteria or fungi. This changed how we understand infectious agents. Next, he identified nitrogen-fixing bacteria, such as Azotobacter crucoccum. This discovery was huge because it gave us new insights into the nitrogen cycle, showing how bacteria help convert atmospheric nitrogen into forms plants can use. It also had a big impact on agriculture, leading to the development of better fertilizers. He also discovered sulfur-reducing bacteria, like desulfuromonas acetoxidans. This helped us understand the sulfur cycle better and revealed how bacteria play a role in processes like the formation of acid mine drainage, which is important for environmental science. All of these discoveries highlight Beijerinck's incredible contributions to microbiology. They not only advanced our scientific knowledge, but also connected to broader themes like the cycles of life and the importance of microorganisms in our environment. Now, let's talk about two more key contributions from Beijerinck that truly transformed microbiology. First, he developed the enrichment culture technique, which is a fundamental method we still use today. Essentially, this involves creating specific conditions in the lab that favor the growth of particular microorganisms from complex environmental samples. This was a game changer because it allowed scientists to isolate and study microbes that were previously very difficult to cultivate. Thanks to this technique, we could explore microorganisms in their natural environments and better understand their roles. Next, Beijerinck's work on symbiotic relationships was equally groundbreaking. He studied how certain soil bacteria interact with plants, especially legumes. He discovered that these plants host nitrogen-fixing bacteria in their root nodules. This finding was crucial because it laid the foundation for understanding how plants and microorganisms form mutually beneficial relationships. Today, this knowledge is vital in agriculture and ecology, helping us improve crop yields and maintain healthy ecosystems. Both of these discoveries highlight how Beijerinck's insights helped us see the microbial world as an integral part of natural processes and human life. They also connect back to our overall theme of understanding microorganisms, vital roles in our environment. Let's take a moment to look at some of the key landmarks and milestones in the field of microbiology. It all began in 1676 when Anton van Leeuwenhoek invented the microscope. This breakthrough allowed scientists for the first time to observe microorganisms directly, which really marked the birth of microbiology as a scientific discipline. Moving forward to the 1860s, Louis Pasteur conducted groundbreaking experiments that challenged long-standing beliefs. He showed that microorganisms do not just appear spontaneously, but instead, they come from pre-existing microorganisms. This was a huge shift in understanding. Pasteur also developed pasteurization, a process of heating liquids to kill bacteria and molds, which has had a lasting impact on food safety. Then, in the late 19th century, the germ theory of disease emerged. Louis Pasteur and Robert Koch independently proved that microorganisms are actually the cause of many diseases. Koch went further with his postulates. These are a set of guidelines that help scientists establish the specific microorganism responsible for a particular disease. These milestones really laid the foundation for modern microbiology and our understanding of infectious diseases. Let's take a moment to reflect on some of the most groundbreaking discoveries in microbiology. In 1928, Alexander Fleming made a discovery that truly changed the course of medicine. He identified penicillin, the first true antibiotic. This was a huge breakthrough because it marked the start of the antibiotic era, fundamentally transforming how we treat bacterial infections. It dramatically reduced mortality rates and saved countless lives. Moving forward, vaccination and immunization have been pivotal in disease prevention. Starting in the late 18th century, Edward Jenner developed the smallpox vaccine, which was a major milestone. This innovation opened the door to modern vaccination practices. Over time, vaccines against diseases like polio, measles, and influenza emerged, saving millions of lives worldwide. These advances highlight how preventive medicine can be as powerful as treatments. And then, in 1953, a discovery in genetics took a major leap forward. James Watson and Francis Crick proposed the double helix structure of DNA. Understanding DNA's structure was fundamental because it laid the groundwork for unraveling how genetic information is stored and inherited, both in humans and microorganisms. This discovery has been essential for advances in genetics, molecular biology, and even the development of new therapies. All of these discoveries, from antibiotics to vaccines to genetic understanding, are interconnected milestones that have shaped modern medicine and public health. They demonstrate how scientific progress continuously pushes the boundaries of what's possible in fighting disease and improving health outcomes. Now, moving on to the 1970s, we saw the development of recombinant DNA technology. This was a game changer because it allowed scientists to manipulate and transfer genes between different organisms. Think about how this opened up possibilities across medicine, agriculture, and industry. It's really transformed those fields. Then, in 1983, Carey Mullies invented the polymerase chain reaction, or PCR. This technique is all about amplifying DNA sequences, which means making millions of copies from just a tiny amount of genetic material. PCR revolutionized molecular biology research and diagnostics because it made detecting small amounts of DNA or RNA much more feasible, speeding up everything from disease diagnosis to genetic research. And since the 1990s, we've been in what's called the genomic era. Advances in DNA sequencing technologies have allowed us to sequence entire genomes, including microbes, giving us deep insights into microbial diversity, evolution, and function. One major project during this time was the Human Microbiome Project, launched in the 2000s, which aims to understand the complex microbial communities living in and on our bodies. These developments are not just technical milestones. They're crucial for understanding health, disease, and the intricate relationship between humans and their microbial environment. In 2012, a major breakthrough happened with the development of CRISPR-Cas9 gene editing technology by Jennifer Doudna and Emmanuel Charpentier. This innovation allows scientists to make precise modifications to DNA sequences, which is a game changer in genetics and molecular biology. What makes CRISPR-Cas9 so groundbreaking is its ability to target specific parts of the genome and edit them accurately. This has enormous potential across various fields, including microbiology, where it enables targeted genome editing in microorganisms. Essentially, it opens up new possibilities for understanding and manipulating microbial genomes, which can lead to advances in medicine, agriculture, and beyond. This slide highlights how this technology fits into the broader theme of genetic innovation and its transformative impact on science and society. Spontaneous generation, also known as abiogenesis, is an idea that has long been discredited, but it's interesting to see how it shaped early thinking. The concept suggests that living organisms could arise from non-living matter under certain conditions. For centuries, this idea was quite widespread, both in scientific circles and philosophical thought. People used it to explain everyday observations, like maggots appearing on decaying meat or mice seemingly popping up in stored grain. In ancient civilizations, such as Egypt and Greece, spontaneous generation was widely accepted. People observed what seemed like living creatures appearing out of nowhere and believed this was natural. These observations, although lacking modern scientific understanding, influenced their explanations of life's origins. However, the controversy surrounding spontaneous generation grew over time, especially as scientists began to test and challenge these ideas. Today, we know that this concept is not supported by scientific evidence. Connecting this to our overall theme, it's a reminder of how scientific ideas evolve, from widely accepted notions to theories replaced by rigorous experimentation and proof. In the 17th century, Italian scientist Francesco Redi conducted a series of experiments that really challenged the long-held beliefs of his time. He focused on decaying meat and flies, which was a common example used to support the idea of spontaneous generation. Redi's key insight was to test whether living organisms could actually arise from non-living matter. He set up experiments where he exposed meat to flies, and in other cases, covered the meat to prevent any flies from reaching it. What he found was quite revealing. Maggots only appeared on the meat that was uncovered and accessible to flies. The covered meat, which was protected from flies, did not develop maggots at all. This was a major breakthrough because it directly challenged the idea of spontaneous generation, that life could just appear out of nowhere. Instead, Redi's work showed that flies and their maggots came from eggs laid by adult flies, not from the meat itself. This experiment is a crucial piece in the story of scientific discovery because it laid the groundwork for understanding that life comes from existing life, a concept that forms the basis of modern biology. It's a perfect example of how careful observation and experimentation can overturn long-standing beliefs and lead us to a clearer understanding of nature. In the 18th century, scientists like John Needham were exploring the origins of microorganisms. Needham conducted experiments where he boiled nutrient broth to kill any existing microbes, then observed that microbes appeared again after some time. He took this as evidence supporting spontaneous generation, the idea that life could arise directly from non-living matter. However, it's important to note that his experiments faced criticism later on, mainly because the controls weren't sufficient. For example, he didn't always prevent contamination or ensure that no microbes from the environment could get in. These shortcomings led to questions about whether his results truly proved spontaneous generation or if other factors were at play. This highlights how scientific understanding evolves and why rigorous controls are crucial in experiments. Overall, Needham's work played a role in shaping the debate about the origins of life, which would eventually lead to more refined experiments and clearer conclusions. In the same century, Lazzaro Spallanzani decided to revisit Needham's experiments, but he made a crucial change. He used sealed flasks, which prevented air from entering. Now, this is important because air can carry microorganisms, so by sealing the flasks, Spallanzani was controlling for that variable. When he observed the results, he saw no microbial growth inside these sealed flasks. This was a significant finding because it directly challenged Needham's results, which had shown microbial growth. Spallanzani's work supported the idea that spontaneous generation was unlikely to occur without pre-existing life. It's a key step in understanding that life comes from life, and it highlights how controlling experimental variables is essential in scientific discovery. This experiment helped shape the future of microbiology and our understanding of how life arises. So, back in the mid-19th century, Louis Pasteur conducted a series of pivotal experiments using a special type of flask known as the swan-necked flask. What made this design so clever was that it allowed air to enter, but prevented particles, like dust or microorganisms, from getting in. This setup was crucial because it let Pasteur test whether microorganisms could spontaneously generate from non-living matter. And what he discovered was quite groundbreaking. His experiments conclusively disproved the idea of spontaneous generation, the notion that living things could just appear out of nowhere. Instead, what he found provided strong evidence for what we now call biogenesis, the principle that living organisms only come from other living organisms. So the key takeaway here is that Pasteur's experiments showed clearly that life cannot arise from non-living material. It can only originate from pre-existing life forms. This became a fundamental concept in biology, shaping how we understand the origins of life. Conti, now, as we move forward, it's important to recognize how these elements interconnect within our overall strategy. The key points here emphasize the significance of consistency and clarity in our messaging. When we maintain a cohesive approach, it not only reinforces our brand identity, but also builds trust with our audience. I want to highlight that understanding these foundational concepts allows us to better adapt to changing market conditions while still staying true to our core values. Remember, the overarching theme of this presentation is about creating a strong, unified presence, and these details play a crucial role in that process. So, let's keep these ideas in mind as we continue to develop and refine our approach. Today, I will discuss the theory of special creation, which is one of the earliest ideas about the origin of life. This theory states that the entire universe was created by a supernatural power, which we refer to as God. It was proposed by Hebrew scholars and gained significant support from Father Suarez. According to this theory, God created the universe in 6 days. On the first day, he created the earth and the heaven. The sky was formed on the second day. On the third day, land and plants came into existence. The sun, moon, and stars were created on the fourth day. Fish and birds appeared on the fifth day. And finally, animals, including human beings, were created on the sixth day. In this context, Adam is considered the first man, and the first woman was Eve. This explanation reflects a religious perspective on how life began, emphasizing divine intervention. As we move forward in this presentation, we'll look at current scientific theories about the origin of life and microbial evolution, which provide different insights from this traditional view. According to Hindu mythology, God Brahma created life in a single stroke. The first man was Manu, and the first woman was Shraddha. However, because this story lacks scientific explanation, it was often rejected by those seeking naturalistic accounts of life's origins. Moving on to the theory of spontaneous generation, or abiogenesis, this idea suggested that living organisms could arise spontaneously from non-living materials like dung, mud, or earth. It was proposed by von Helmont and supported by thinkers like Anaximenes and Aristotle. This theory explained that insects, for example, could originate from dew, frogs and toads from muddy pond bottoms, maggots from decaying meat, tapeworms from animal excreta, and microorganisms from air or water. But as scientific methods advanced, many experiments challenged this idea. Riddi in 1668, Pasteur in 1864, and Spallanzani in 1765 all conducted experiments that ultimately rejected spontaneous generation. Their work showed that life does not simply arise spontaneously from non-living matter, paving the way for our current understanding of biogenesis. The slide proposes that life arises only from pre-existing life, a concept known as biogenesis. This idea is supported by several experiments, and one of the most famous is Riddi's experiment from 1668. Francisco Riddi, an Italian physician, took a piece of boiled meat and placed it in 3 separate jars. He covered one jar with parchment paper, another with muslin cloth, and left the third jar open. After some time, he observed that maggots only appeared in the open jar, where flies could lay eggs directly on the meat. The jars covered with parchment and muslin did not develop maggots, indicating that flies, and not the meat itself, were responsible for maggot formation. Riddi concluded from this that life, specifically maggots, can only come from other living things, flies in this case, and not from non-living substances. This experiment was a strong challenge to the idea of spontaneous generation, and supported the idea that life arises only from pre-existing life, which is the backbone of the biogenesis theory. It's a key point in understanding the origins of life and how we see the natural process of reproduction and growth. In this experiment, I used 8 bottles filled with hay infusion, which is a nutrient-rich liquid encouraging microbial growth. I then boiled the bottles to sterilize the contents, ensuring that no pre-existing microorganisms remained. After sterilization, I kept 4 of these bottles airtight, sealing them tightly, while the other 4 were loosely corked, allowing some air to enter. Over the next few days, I observed what happened. The loosely corked bottles developed a dense growth of microorganisms, clearly visible as cloudy or fuzzy masses. In contrast, the airtight bottles remained free of any microbial growth. This experiment led to a crucial conclusion. The air itself must contain microorganisms, and these tiny life forms are the source of contamination. It supported the idea that microorganisms come from the environment, not spontaneously generated within the sterile liquid. This was a key step forward in understanding that air carries these microorganisms, which is fundamental to the broader concept of germ theory and the importance of sterilization in preventing contamination. In 1864, Louis Pasteur, a French biochemist, conducted a famous experiment to support the idea of biogenesis, which states that life comes from pre-existing life. He used a special flask with a neck bent into an S shape, which he heated to make it flexible. Then, he filled the flask about halfway with a nutrient solution, which provides the necessary substances for microorganisms to grow. After that, he boiled the entire flask for several hours to kill any existing microorganisms in the solution. Once boiling was complete, he sealed the neck of the flask and left it undisturbed for several days. During this time, no signs of life appeared in the flask, which was quite significant. But here's the interesting part. When he broke the neck of the flask, microorganisms suddenly appeared and started to grow. This experiment was crucial because it showed that microorganisms do not spontaneously generate, but come from pre-existing life. It was a key step in proving the principle of biogenesis and understanding that life arises only from other life. This theory, known as the Cosmozoic theory, proposes that life did not originate on Earth itself, but rather arrived from outer space. The idea is that spores or seeds of life, called panspermia, traveled across the universe and somehow seeded life on our planet. Historically, figures like Richter in 1865 and later Arrhenius supported this concept, suggesting that life could have been carried here by meteorites or comets. However, there are significant challenges with this theory. For example, it's difficult to explain how these spores could survive the extreme conditions of space, including the intense heat during entry into Earth's atmosphere, as well as harmful radiation. These harsh conditions make the survival and transfer of life forms across space quite problematic. Despite these difficulties, the idea highlights an intriguing possibility that life on Earth might have extraterrestrial origins. It also connects to broader questions about the universe and the potential for life beyond our planet, which continues to inspire scientific exploration today. According to this theory, around 4.2 billion years ago, life began to emerge in water on primitive Earth through a series of chemical reactions. This idea is what's called the biochemical theory of the origin of life. It's often referred to as the modern synthetic theory because it combines chemistry and biology to explain how life could have started. This theory identifies three main stages in the process. First is chemogeny, which is the chemical synthesis of organic compounds. Then comes biogenesis, where these compounds organize into simple living organisms. And finally, cognogeny, which involves the development of more complex life forms from these early organisms. So, in essence, this theory links the chemical processes happening in Earth's early waters to the eventual rise of life, connecting chemistry directly to biology. It highlights how life's origins are rooted in natural chemical reactions, making it a key piece of our overall understanding of life's beginnings. Microbes were actually the very first life forms to appear on Earth, and since then, they have continued to evolve and diversify at a remarkable pace. Their evolution is driven by the same fundamental forces we see in other organisms: natural selection, genetic drift, and mutation. But what makes microbial evolution particularly fascinating and dynamic is their unique features. One key aspect is their high reproductive rate. Microbes can produce huge numbers of offspring in a very short amount of time, which significantly increases the chances of mutations occurring. Mutations are the raw material for evolution, after all. Additionally, microbes have a special ability called horizontal gene transfer, which means they can exchange genetic material directly with each other. This process enables them to acquire new genes and traits rapidly, helping them adapt to changing environments or new challenges. All of this ties into our broader theme of evolution, showing how adaptable and resilient microbes are, and highlighting the incredible speed at which they can evolve. It's a reminder of just how dynamic life on Earth really is. Microbial evolution has truly been a driving force in shaping our planet's biosphere. For instance, microbes were pivotal in the oxygenation of the atmosphere, a process that fundamentally transformed Earth and made it possible for complex multicellular life to develop. Without these tiny organisms, the world as we know it would be very different. But their influence doesn't stop there. Microbes are essential players in nutrient cycling, breaking down organic material and recycling vital nutrients that support all life forms. They also contribute to biodegradation, helping to clean up pollutants and maintain ecosystem health. Overall, these microbial activities are integral to the stability and diversity of life on Earth, linking back to our broader theme of how microbial processes have shaped and continue to sustain our planet's ecosystems.